Hello, so these are the instructions to go along with the Biology 120 Lab 11. In this lab, we're starting to look at the diversity of animal groups. So we're covering a lot of different groups today. We're looking at the sponges, the Cnidarians, and then three groups of protostome worms, groups that are traditionally been referred to as worms, the flatworms, roundworms, and segmented worms. So one good way of studying for the lab exam questions that will deal with this lab is to go through the lectures. So go through the lectures where I talk about these animal groups. That'll give you the general characteristics for each group, as well as I have a lot of pictures and videos of live animals. So that'll help you with questions on the lab exam that ask you, for example, to identify a living specimen, or a preserved specimen, like what animal grouping would this belong to? So for those general characteristics, what defines flatworms, what separates them from roundworms and segmented worms, how can you tell them apart? Look through the lecture. So this presentation is going to focus on lab-specific materials, so content you would have seen in the lab if we had the meeting. So especially slides and then the information about the two dissections that we were scheduled to do in this lab. So let's start with sponges. So again, look through the lecture material. When I talk about sponges, know general characteristics and anatomy of sponges, and be able to recognize a sponge as a sponge. So if I showed you on the final lab exam a picture or a video of a sponge, you would know what it was. And the main thing for this, so I'm showing you here a few examples of sponge slides that you should recognize sponge slides as coming from a sponge. So you don't actually have to be able to differentiate microscopic structures on the slide. The main thing is if you saw a slide of a section through a sponge, you would know what it was. So if we look at this slide here that I've posted onto this presentation, you can see in the middle of the sponge there is a hollow space, which I'm going to circle here, called the spongiocele, and that's where the waste products would go, and then eventually they would leave the top of the sponge, called the osculum. But in order to get the osculum, you would have to slice through the osculum. So in this case, we're just slicing through the middle of the sponge, so we don't have the osculum there. There are openings, pores, on the outside of the sponge. So that's what you see here. These are pores. And eventually the water and food would go into these circular chambers. They're actually called flagellated chambers. And the flagellated chambers are lined by those special sponge-specific cells, the choanocytes, the collared cells. So here are some more sponge slides showing you these structures on a closer scale. So here we can see this is one flagellated chamber. You can see the cells lining this chamber would be the choanocytes, the flagellated cells. And when it's preserved like this, you can't necessarily see the flagella on them, but you can see these open chambers lined by cells. Here's another view here on the right. You can see these choanocytes lining these flagellated chambers. And the choanocytes would have flagella, and they would capture food particles on their collar. Another kind of like distinguishing feature of a sponge slide that would help you recognize what you're looking at as a sponge slide are these spicules. Hopefully you remember from the lecture that sponges, their basic body structure, their body outline is made up of these little microscopic shards called spicules. And the spicules can be made of different materials, like there's glass spicules. There's also chalk spicules made of calcium carbonate. That's what we have here in this example. So this is a chalk sponge. There are the soft sponges that are made of spongin, the kind of spongy protein. But this would be a chalk sponge. So if you look at it under, in this case, 100 times magnification, you can see these, they look like little tiny glass shards, little fragments of glass. In this case, they're actually chalk, not glass, but that's what the spicules look like. So this would be kind of present throughout the outline of the sponge's body. So sometimes it gets lost, but on the living sponge, you would have spicules kind of like within the framework of the sponge's body. So then we turn to the Cnidarians. And for the Cnidarians, again, from the lecture, try to learn and remember the general characteristics of Cnidarians, that they have their two body forms, the polyp and medusa, they have the stinging cells, the cnidocytes, they have radial symmetry. And also on the exam, the lab exam, be able to recognize a Cnidarian as a Cnidarian. So if you saw a picture or a video of a jellyfish or a hydra or a sea anemone or a coral, you would be able to say, I know what that is a Cnidarian. That belongs to the Cnidarian group. So for this particular lab activity, what we were focusing on for the slides was hydra, which is a freshwater polyp species of Cnidarian belonging to the hydrozoan group. So it just kind of has this polyp stage. And for hydra, they frequently reproduce asexually by budding. So on this picture on the right, you can see there was the original polyp hydra, and then it has a little tiny asexual bud coming off the side. And it's considered budding because it's creating offspring asexually that are quite a bit smaller than the original adult. So this would eventually break off and then go and grow into a full-sized hydra. So these are called whole mounts, where you basically just take the whole organism and put it on a slide. So just be able to recognize, that this is hydra and also this process of budding, if you saw this on the exam. We also have sections through, a longitudinal section through the body of a hydra. So that means you're slicing the hydra lengthwise. So on this left picture here, you can see the tentacles here at the top. So it's sliced through a couple of tentacles. So hydras, like Cnidarians in general, have a sac-like gut. So there's one mouth opening, and that will lead to their gut. And their gut is just a chamber with one entry and one exit. So food goes in through the mouth, and waste also exits the mouth. So it's not a tube-like gut, it's a sac-like gut. Okay. Hydra also has a very, very thin body surface. So their whole outer body wall is made up of two cell layers. There's the epidermis on the outside, which you can see here close up. That's the epidermis. And then this inner cell layer that kind of looks like little fingers and has a lot of surface area is the gastrodermis. And that would line their gut cavity. And this is where they would be digesting and absorbing food. And because they just have these two cell layers, food will just move to the epidermis by diffusion. But yeah, so they just have these two cell layers that make up their body wall, and also just kind of keep in mind that they have this sac-like gut. So that's the takeaway from these slides. Then we look at the groups that are usually referred to as worms, although they're not closely related to each other, starting with the flatworm. So know the general characteristics of flatworms from the lecture PowerPoints. And then recognize a flatworm as a flatworm. So if you saw Dugessia, which is a non-parasitic planarian, be able to recognize that as a flatworm, or if you're looking at flukes or tapeworms, you would know those belong to the flatworm group. The tapeworm has a pretty cool slide that, it's unfortunate we missed out on, but I've tried to replicate it here. So on the tapeworm slide, what they do is they cut the tapeworm's body into different body regions, because the tapeworm's body is pretty long. So they slice it up into different regions and they put representative regions onto that slide. So on the left here, actually both of these pictures are pictures of the scolex of a tapeworm. This is the top of the body. Not exactly a head, because they don't have a mouth or a brain or sensory organs, but it is the top of the body. And this is what they would use to attach to the intestine. So you have these nice hooks on the end that would dig into the intestinal wall, as well as the suckers, 4 suckers underneath the hooks. And same thing here on the left, I just gave you two different views of tapeworm scollexes. As you go down the body of the tapeworm, so past the scollex, you have the segmented body regions. Their segments are referred to as proglottids. And I think I mentioned this in the lecture, but kind of a funny thing about the tapeworm is that the proglottids close to the scollex, close to the top, are reproductively immature. So if you look at this picture on the left, this is what they look like. They just kind of look empty. They don't have reproductive organs at all. They have not developed yet. So this is what it would look like close to the scollex. And as you go down and down the long body of the tapeworm, the proglottids will become reproductively mature, and they'll have eggs in them. So this would be a mature proglottid on the right. You can see all of this little tubing here is the reproductive tract of the tapeworm, and it would be filled with eggs. And to reproduce, the proglottids at the end of the tapeworm just break off, and they just go out with the feces to hopefully, from the point of view of the tapeworm, infect a new host. So now let's talk a little bit about roundworms. So for roundworms, again, know the general characteristics of roundworms and be able to recognize a roundworm if you saw one, that yes, that would be a roundworm. They have a round, unsegmented body, a complete digestive tract, so they have a separate mouth and anus. For this lab activity, we were going to dissect Ascaris, which is a parasitic species of roundworm, a common parasite of humans. So firstly, with this parasite, you should know the external anatomy of the parasite. And in this case, the only thing that I really want you to pay attention to, because there's not a lot of features on the Ascaris, is be able to tell the difference between a male Ascaris and a female Ascaris. So as you can see in this picture on the left, the female is a lot bigger, a lot thicker around than the male. Male's much smaller. You can also see that on the right here, female's thicker and longer than the male. The other way that you can tell which is on the posterior end of the male, there's a little hook. There's a hook at the end, and he uses that to hold on to the female when they mate with each other. The female doesn't have a hook, so if you look at both ends of the female, they are just straight. So if you saw a thinner, smaller Ascaris that has a hook on one end, that's a male. So for the Ascaris dissection, they are obligate parasites, so they do not have a ton of different organ systems inside their body. There's not a lot to see. They are mostly a bag of reproductive tissue. So inside their body, they do have a pretty simple digestive tract made up of this intestine. So you can see the intestine I have highlighted in both of these dissection photographs. The intestine looks like a flat, thin piece of tape. So it's very, very thin and delicate. You can see that there. It just runs along the length of the roundworm's body. So that's the intestine. And then the other feature to see is the reproductive parts. And so, you know, you don't have to know too much here as far as the final exam. The big thing is that you would be able to tell if it's male or female from looking at a dissected Ascaris. And it's not that hard. So the best way to tell if it's a male or female, you look for this thickened part of the reproductive tract. So you can see I highlighted here. This picture is a little blurry, but I couldn't find a better one that shows this to you. So this top part here that's flat, that's the intestine I showed you before. But this lower tube that kind of looks a little bit windy, that's the seminal vesicle. That's part of the male reproductive system. And you can tell this is a male because it just has one thick part, okay? You can look at this picture on the right. They basically pulled out the male reproductive tract at the end here. This is where it would be a seminal vesicle. It would be thick. There's just the one tube, one thick tube. So if you saw this, if you cut an Ascaris open and you saw one thick tube running next to the intestine, that would tell you you're looking at a male. Okay. So this will become more clear when we compare this to a female. So this is the inside of a female Ascaris, and I found quite a bit better pictures for this one. But the way you can tell it's a female is there are two thick parts, two thick tubes that would run alongside the intestine. And so this is the two parts of the uterus. Okay, so you would have two thick tubes running alongside each other and then also alongside the intestine, which is down here. Again, in this other picture, you can see here's one of the thick tubes, here's the other thick tube. So that's both the two parts of the uterus. So that's how you could tell pretty easily if you look at the inside of an Ascaris, are you dissecting a male or a female? You can also tell this from the external anatomy, but it's just another way that you could differentiate the two is just look at those reproductive parts. And do you see one long tube that's thick, or do you see a couple of them running alongside each other? Okay. Then we get to the segmented worms, Annelida. So again, know the general characteristics for the segmented worms. Be able to recognize a segmented worm as a segmented worm. So if you saw an earthworm or a leech or a polychaete, that you would be able to recognize those as segmented worms. So the dissection for this part of the lab was an earthworm dissection. And I always like to start with, the external anatomy, what you should know as far as external anatomy. So one of the big features when you look at a segmented worm, well, you can tell they're segmented. They have these different, little lines across the body. But they also, if they're reproductively mature, they have this structure that kind of looks like a band-aid, part of the way down their body, closer to one end. That's called the clitellum. That's used in reproduction. That actually will secrete a slimy covering that will become a cocoon that houses the eggs when they reproduce. That's secreted by the clitellum. The clitellum is closer to the head end, to the anterior end. So if you're looking at an earthworm and you see the clitellum, that'll tell you, the end closer to the clitellum is the head end, the anterior end. The end opposite would be the posterior end. Anterior end would have the mouth. Also, most of the interesting anatomy is closer to the head end. The posterior end has the anus. So again, in this picture on the left, that would be the anterior end, close to the clitellum, and this back here would be the posterior end. Otherwise, it's sometimes hard to tell because they can kind of move in either direction. So that's the best way to tell if you're looking at anterior or posterior. So now let's look at the actual dissection of the earthworm. So there's a few things that you should know for the earthworm dissection. I'm first going to talk about the digestive system of the earthworm. There's just 4 structures that I would want you to know as far as the final lab exam. But first, I'll talk about the whole pathway that food would take, because some of these structures you don't have to know. So there's the mouth. The mouth would be on the end, the anterior end of the earthworm. After the mouth, food gets sucked into the pharynx, which is kind of a thick muscular organ that's strong and powerful, and it uses that power to suck food into the digestive system. After the pharynx, there's a kind of a thin tube called the esophagus. It kind of runs alongside here, but it's difficult to see, so I don't actually test on that one. But after the esophagus, food goes into the crop. The crop stores the food, kind of like a sack. And then after it's been stored for a while, the food gets passed to the gizzard, which is right next to it. And the gizzard is a muscular organ. It's a muscular stomach that churns the food to help break it down. And then after the gizzard, we have the intestine. And the intestine will travel all the way down the body of the earthworm until food exits, the waste exits out the anus. So that's the complete pathway of the digestive system for the earthworm. So what I would actually want you to know are the structures that I think are pretty easy to tell apart. So right after the head, you have the pharynx. Pharynx looks like kind of a thick region. So here it is in this picture here. There's the pharynx. Pharynx, and this one's a little bit harder to see, but it would be right there. Esophagus, difficult to see, so you don't have to recognize it on the final exam. But then these 2 structures are easier to see. So there's the darker, usually darker crop. So the crop would be this first region right here. And then that leads into the lighter structure, which is the gizzard. And the gizzard, if you pressed on it, would feel really hard because it's muscular. So let's look at it in these other pictures. So here's the crop here. This would be the crop. And then this wider structure here would be the gizzard. This one's labeled, so you can kind of clearly see there's kind of the separation there. So this would be the crop, and then this other lighter structure would be the gizzard. And then after the gizzard, you just have this long tube, which is the intestine. So you should also know that. And you've got the intestine labeled in both of these drawings. Intestine starts right after the gizzard and goes all the way down to the anus. So those are the 4 structures you should know for the earthworm's digestive system: pharynx, crop, gizzard, intestine. Not too bad. There's 2 more structures I want you to know for the earthworm. So they have 5 rows of hearts. They're also called aortic arches. So that's part of their circulatory system. That's how they would pump blood throughout the body of the earthworm. And they're sometimes a little bit hard to see, but it might be better to start with the other structure that you should know, which are the seminal vesicles, and those are used in reproduction. They send sperm to the other earthworm when they mate. Earthworms are hermaphrodites, so they line up end to end and they mutually impregnate each other. So the seminal vesicles, that's these larger, kind of creamy looking structures here, close to the top of the head. Okay, they will send sperm out to the other earthworm, and at the same time they'll be receiving sperm from their partner, and the sperm will be collected in these structures, which are a little bit smaller, called seminal receptacles. They're a little harder to see, so for testing purposes, I just want you to remember the seminal vesicles, which are usually pretty obvious. So in this picture here, you can see those are the seminal vesicles. These are the seminal vesicles here. So they're more posterior compared to the seminal receptacles, and they're larger. So those are seminal vesicles used in reproduction. Usually close to them and a little bit above them would be the hearts. You might not see all 5 bands, but they kind of look like little dark lines that cross the earthworm. So there would be one. Here's another one in this photograph. Here's another one. And here's another one. I just see these 4 clearly, but they would have 5. In this picture at the bottom, they injected the circulatory system with pink dye. So you can see there's one of the hearts going across there. There's another heart. This one's difficult to see, this top one. I think they didn't inject any dye into the circulatory system, so hearts are difficult to see. But if you just kind of remember it as like these dark bands that are around the seminal vesicles, those would be the hearts. So that's all of the structures that I want you to know for the earthworm dissection, and that takes us to the end of this lab. So I will post this as a PDF onto the Canvas page, and feel free to watch this video again if you need to. And I will also post an assignment report to go alongside this that will ask some questions to just make sure you're kind of learning the material correctly. Okay, so I'll see you again in the next video.